Let's move on to the ear now, and specifically the first learning outcome for the ear, which is describe the three divisions of the ear. Here we have a cross-section of the ear, as if we've taken a cross-section along the frontal plane. We have three divisions of the ear. The external ear, which is made up of the auricle or pinna, the portion that we would tend to call the ear in everyday language, and then the external auditory canal. Here we have the tympanic membrane, or what we would call the eardrum. This is the division between the external ear and the middle ear. The middle ear is this region here in the middle that is made up of these three bones called ossicles. The term ossicle just means small bone. And the three small bones, the smallest of your body, are the malleus, the incus, and the stapes. What you can see here is that the malleus is actually attached to the tympanic membrane, and this will be important when we look at how the sound actually moves through the ear. The stapes on the other end then is connected to the inner ear. The inner ear is made up of three different regions. So starting on the left, we have the semicircular canals. Semicircular, meaning partially circular canals, and that's because they do run in somewhat of a circle, extending off the second portion here, the vestibule. The vestibule is actually where the stapes attaches to the inner ear. It also plays a role in balance that we'll look at in a few moments. Finally, we have the cochlea. Cochlea gets its name from snail, and you can see that the cochlea has a sort of snail shell shape to it. Finally, we have the cranial nerve number 8. This is the vestibulocochlear nerve, because the vestibular nerve is coming off of the vestibule, and the cochlear nerve is coming off of the cochlea, and then they connect together to form the vestibulocochlear nerve, cranial nerve number 8. How does our ear actually translate sound into nervous impulses? Let's look at the steps in hearing in order to understand this process. Let's start by talking a little bit about sound. Here we have a speaker, and the reason I put this in the slide here is to show you how a speaker actually works. The speaker is moving in slow motion, and ultimately the speaker is actually vibrating, moving back and forth. That's because sound is created by movement of air particles. So in order to create the sound of the music that's playing in the speaker, we actually have to vibrate air particles. The speaker is actually moving back and forth in a way that if we were to actually look at the air particles in front of the speaker, we'd see something along these lines. Every time the speaker vibrates, it bumps into the air particles that are directly up against it. That sends the air particles moving forward, bumping into the next particles over, which then bump into the next particles over, and so on. But what you can see here on the right side is that we actually then have waves of sound, waves of particles bumping into each other that then transmit or move through the room. Any sound ultimately that you hear is the movement of air particles. If I were to hit my hand on the table, the reason that you hear that sound is because as I hit the table then starts vibrating, just like the speaker is here. It starts bumping up against the air that's next to it, and then, just like in the image here, those particles bump into the next particles over, and they make their way to the microphone that I'm currently using. In the sense of this recording, the sound information is then translated into electronic information, and then when you listen to the presentation, the earphones convert that electronic information back into sound moving air particles that you then hear. So to continue and actually list the steps in hearing, let's start with some sort of sound. This person here is playing guitar and singing, so they're producing sound waves. Those sound waves will transmit through the air, as we just saw in the previous slide, and they will make their way to the pinna, to the auricle, your outer ear, and then that pinna works as sort of a funnel to actually collect those sound waves and transmit them through the external auditory canal down towards the tympanic membrane. Because those sound waves are vibrating particles, when they hit the tympanic membrane will actually start vibrating. The ossicles then, the malleus was attached to the tympanic membrane, so as the tympanic membrane starts vibrating, the malleus will vibrate. As the malleus starts moving, the other two ossicles will start moving because they're all connected. As the stapes starts moving, it's attached to the inner ear, and it will start moving the inner ear. The cochlea is the portion of the inner ear responsible for translating sound into action potential along neurons. In order to look at how it does that, let's take a cross-section of the cochlea. The cochlea looks like this, where we have a bony outer material with 3 compartments. Those compartments are actually filled with fluid. So the outer 2 compartments are filled with perilymph. Inside the inner compartment, we have endolymph. The endolymph fluid starts moving because the cochlea is being shaken by the stapes. Here we can see the organ of Corti, which is a region with sensory receptors. And if you look closely, those sensory receptors have little hair-like extensions coming off of them. As the cochlea vibrates and the endolymph moves within this region, the endolymph actually presses up against those hairs of the receptor cells. As those hairs are pressed upon and move, they actually trigger an action potential on a sensory neuron that you can see beneath them that then runs out along the cochlear nerve to the vestibulocochlear nerve. Thus, we've translated movement of air particles in terms of sound waves into an action potential in this region of the organ of Corti. The ear also plays a role in equilibrium. So let's take a look at the inner ear and the various regions here. So we've already mentioned the cochlea on the right side that plays a role in turning sound into the action potential along a sensory neuron. If we take a look at the left-hand side here, we have the semicircular canals and the vestibule. We'll start by looking at the semicircular canals. As you can see, there are 3 semicircular canals, and they run in 3 different directions. As we can see on the left side, there's 2 layers of the ear. We have the bony labyrinth, contains perilymph, so we saw that earlier when we were looking at the cochlea. And then we have the membranous labyrinth, which contains endolymph, just like we saw when we looked at the cochlea earlier on. We have the semicircular canals here that have very similar structure in terms of the types of fluid and the types of containers, those labyrinths that make up the semicircular canals and indeed the entire inner ear. If we were to zoom in on the region shown here, what we'll see is that there are actually receptor cells that are hair cells, just like we saw over in the organ of Corti. There are these long processes of the hair cells, and ultimately these semicircular canals are responsible for dynamic equilibrium. Let's think of an example. If I were to be sitting looking straight ahead and then someone called my name off to the left, I'd quickly turn my head. My ears would of course turn with me, but because these semicircular canals have the endolymph within the membranous labyrinth, that fluid undergoes inertia. The fluid does not want to move, and so as I move my head, it takes a moment for that fluid to actually get moving, and therefore the fluid will press upon the processes of these hair cells, and that will ultimately cause an action potential along the sensory neuron, sending information to my brain that my head is turning. Therefore, my brain now can help maintain my balance so I don't look to the left and then fall over because I'm all thrown off, but my brain can actually help coordinate my muscular movements so that I maintain my proper position and I don't fall. This is dynamic equilibrium because it's involved in quick motions. To think of another example of dynamic equilibrium, think about maybe when you were a kid and you like to play with your friends and maybe you'd stand and spin around in order to get dizzy. What's going on in that situation is as you spin, the fluid, the endolymph inside the membranous labyrinth starts to actually get moving. And you're spinning and all of a sudden you stop. But what ends up happening is that there's inertia for the endolymph, and the endolymph keeps moving even though you've stopped. And so it continues to press upon these processes, sending information to your brain that you're spinning. And so you end up feeling dizzy because your brain is getting this information that you're still moving. Eventually the fluid stops and you go back to your normal state and you have balance. Sometimes we feel dizzy when we get sick. Maybe you've had an inner ear infection before. It would be this region of the ear that's actually infected. As that infection proceeds and we have a buildup of cells and fluids from our immune system trying to attack that disease, we actually have changes in the consistency of the fluid, and that ultimately affects our inner ear, here the semicircular canals, and we end up feeling dizzy because the fluid is not moving as it normally does. Lastly, we have the vestibule. The vestibule is responsible for static equilibrium. The nice part here is the vestibule works in, again, almost the exact same way as the semicircular canals and the cochlea. Within the vestibule, there's fluid, and there are also hair cell receptors. Static equilibrium is different from dynamic in the sense of it's not about movement, but sending information to your brain about the position of your head under static condition or not moving conditions. So if I'm sitting looking straight forward, my brain knows that because my hair cells are not being pressed upon within my vestibule. If I were to look down at my shoes, then the fluid in the vestibule would move, pressing on the hair cells, and again, causing an action potential along the sensory neurons that will run to the vestibular nerve and then to the vestibulocochlear nerve, telling my brain the position of my head, not in the sense of moving, but in the sense of now that I have moved, what am I looking at? Which direction is my head pointed?